In a randomized trial for newly diagnosed breast cancer patients, adding 25 grams of daily ground flaxseed to their diet, delivered inside a simple muffin, measurably altered the biological behavior of their tumors. This was a rigorously controlled clinical trial conducted at the Princess Margaret Hospital and its Campbell Family Cancer Research Institute. Researchers tracked the patients from the day of diagnosis to the day of surgery. On average, the metabolic shifts within the tumor tissue occurred in just 32 days. The biopsy data showed two immediate reactions. First, the Ki-67 index, a primary marker for cellular proliferation, dropped by 34%. Simultaneously, the rate of apoptosis, the programmed death of those cancer cells, increased by 31%. How does a common seed orchestrate this kind of precise, targeted regression in aggressive, hormone-dependent malignancies? This clinical performance stems from two distinct molecular channels. Understanding how flaxseed suppresses tumor growth requires mapping the specific pharmacokinetics that drive them. The first mechanism relies on a class of polyphenols called lignans. Flaxseed is the richest known dietary source of a specific lignan known as secoisolariciresinol diglucoside, or SDG. But SDG has a major biochemical caveat. In its raw form, locked inside the seed, it is biologically inert. Human cells cannot utilize it directly. To become active, the seed must reach the large intestine. There, the patient's colonic microflora must metabolize the SDG into two mammalian enterolignans, enterodiol and enterolactone, or ENL. Once synthesized by the gut bacteria, ENL enters the bloodstream and circulates until it encounters hormone-sensitive tumor cells, which are studded with estrogen receptors alpha and beta. Because its molecular structure is similar to estrogen, ENL acts as a selective estrogen receptor modulator. It physically occupies these receptors, creating a blockade that prevents the body's actual, more potent endogenous hormones from binding and feeding the tumor. Table 7 from the research literature summarizes this evidence. The clinical trial row confirms that this competitive binding action actively lowers estrogen levels and reduces tumor growth in postmenopausal women. This means the anti-cancer efficacy of flax lignans is not innate to the seed itself. It is strictly dependent on the health and composition of the patient's own colonic microbiome. The second pillar of the flaxseed matrix is alpha-linolenic acid, or ALA, a plant-based omega-3 fatty acid. Unlike lignans, which act on external receptors, ALA molecules weave directly into the lipid rafts of the cancer cell's membrane, physically altering its structural dynamics. By changing the membrane's fluidity, ALA initiates a physiological cascade that systematically downregulates the tumor's internal survival wiring. First, it halts the PI3K-AKT pathway. This cuts off the aberrant signals instructing the cell to rapidly survive and proliferate. Second, it suppresses the NF-κB cascade, which drives chronic inflammation. Without that inflammatory signal, the surrounding vascular endothelial growth factors, or VEG, wither, choking off the blood supply that the tumor needs to expand. Looking at Table 5 from the clinical literature, we see the established mechanisms of omega-3 fatty acids, focusing here on the PI3K and NF-κB pathways, which map directly to reduced cell proliferation and lower tumor-promoting inflammation. Together, they form a coordinated dual threat. The lignans starve the tumor of external hormonal fuel, while the ALA dismantles its internal survival and inflammatory wiring. Historically, clinical focus centered almost entirely on the oil. In the mid-20th century, the Budwig protocol attempted to treat cancer patients by combining high doses of isolated flax oil with sulfur-rich proteins like quark. But modern pharmacokinetics reveals a severe complication. To achieve its maximum anti-inflammatory effect, the short-chain ALA and flax oil must be elongated and desaturated into a longer-chain molecule, EPA. This physiological conversion requires the ALA molecule to pass through two specific enzyme gates, delta-5 and delta-6 desaturase. Here is the critical oncological roadblock. Cancer cells frequently lack these exact enzymes. Their metabolic machinery is broken. Without the desaturase enzymes, the isolated ALA pools up at the blockade. It cannot convert into EPA, leaving it trapped in a highly pro-inflammatory state. This creates a genuine clinical risk. Administering large doses of isolated flax oil, especially while a patient is undergoing conventional treatments like radiation, can inadvertently flood the tissue with trapped pro-inflammatory fats, exacerbating the cellular damage it is meant to heal. Relying solely on isolated flax oil ignores the compromised metabolic capacity of the malignant cell. Replicating the success of clinical trials requires a shift back to the whole seed. The success observed in modern clinical trials stems from delivering the complete seed matrix rather than extracting its individual parts. The whole seed retains the lignans that commercial oil extraction strips away. These lignans execute their hormone-blocking function entirely independent of the broken desaturase enzymes. Furthermore, the whole seed delivers the dietary fiber necessary to feed the colonic microbiome, ensuring the bacteria have the raw material to synthesize the enterolignans. The pronounced tumor regression we see in the data is the direct result of this synergy. The enterolignans create a hormone blockade on the outside of the cell, while the baseline ALA suppresses proliferation pathways on the inside. Based on the existing data for both oncoprevention and adjunct therapy, the clinically validated protocol requires 25 to 50 grams of whole flaxseed ground freshly every day to prevent oxidation. However, a significant clinical variable remains. Because conversion depends on flora, the bioavailability of these compounds will naturally fluctuate from patient to patient. To maximize flaxseed's therapeutic potential, the future of precision oncology must involve microbiome profiling to verify that every patient possesses the specific flora needed to synthesize these metabolites.